All right, folks, so we're going to start talking about Chapter 2 today, and I want to kind of make sure we only cover maybe half of the time that we have on Chapter 2, and the rest you're just going to need to kind of make sure that you get caught up on your own, right, with your information, but we're going to cover some of the of Chapter 2, okay? So let's just talk a little bit about this, right? Because I know many of you are already familiar with this, and this is something that we've discussed before, for example, in A&P, or if you've taken a biology class, everything starts with chemistry, right? And we know what atoms are, right? Atoms are the smallest unit of an element, right? And again, makes up pretty much everything that we call matter, right? And that we know that the center of an atom is a nucleus, and that center of an atom contains both protons and neutrons. These are positively charged particles. This is a no charge particle, right? We call that a nucleus, or the center portion of an atom. Surrounding an atom, if you will, are other particles we call electrons. And electrons, of course, are negatively charged. And the positives and negatives basically interact with each other. Depending on how many protons, neutrons, and electrons you have, that gives you all of the different elements in the periodic table of elements, right? Very simple to understand. Many of you already, again, this is a review for many of you. What we see here is a carbon atom, right? And a carbon atom, we know how we have 6 protons, 6 neutrons, and then, of course, we've got 6 electrons, right? And that's what we see there, right? All right. And again, elements are organized by their atomic number officially, right? The number of protons in an element is its atomic number. Okay, it's atomic number, and we organize every single element on the periodic table from 1 to, I think we're at 140 right now, or so far, elements based on their proton number, right? That's the periodic table. You don't need to know anything from the periodic table, right? And we've added, for example, 1 proton or 1 neutron, let's say neutron, to the actual element itself, right? Thereby changing the number of, let's say, neutrons of the element. And when you do that, you then create a different form of the same element. We call that an isotope. Okay? Changing the number of neutrons. Okay? When you change the number of, in fact, electrons, you then, in fact, create a different form of the same element. We would call that ions. Okay? Both of them exist in nature, just by nature. And so we're able to study these, in fact. And one of them that is really important are ions, in fact. Both of them are, but ions, in fact, are really important. When we talk about, for example, bonding or the relationship between atom to atom. Okay? So in talking a little bit more about ions, that's the what? Unequal number of protons and electrons, which means that we are either losing or gaining electrons, changing the element slightly. Still considered the element. Okay? Like sodium, for example. That would be considered sodium. But in fact, if we were to lose an electron here in this element, we would designate it as this. Right? Which you've seen before, I'm sure, chemistry, biology. This is sodium in its natural state because it loses an electron. Okay? Or I can write many other ones just like this. Right? And those all really mean I have gained or lost an electron. Okay? The number plus the plus or the minus on the number indicates how many electrons I lost or gained, and the positive represents how many electrons I've lost, and then the negative basically means how many electrons I have gained. Okay? The reason I talk about this because we talk about bonds here very shortly, and we see that, right? We have names for that, right? So if you're a positive, right, positive ion, you're considered a cation. If you're a negative one, you're an anion. Okay? So here's what we mean. This is like real light situation here that you would see here. This is chlorine, neutral, stable. Well, not stable. It's gained an electron and become the Cl minus, which means an electron, a negative charge has been gained and it becomes a minus. Same thing with lithium there. Lithium loses an electron and becomes a cation, Li plus. Okay, we're just seeing the electrons being given away and the other electron that's been given away being accepted by another element, essentially. Okay? Again, when you vary the number of neutrons, we would create an isotope, and that's what that is as well. And you can create isotopes. For example, there is different types of carbon isotopes, and that's what you see here. This is what exists in nature, so many times we will change the number of neutrons of carbon, and this becomes this number here, C12, C13, and C14. Now, it says 99% of all the carbon that we have on Earth is C12, which is the stable one. We have less than 1% of C13 and less than 1% of C14. What happens with a lot of these on Earth, just to kind of on the side note, because this is all, it's like general bio and geology kind of thing, is that carbon, when stuck in the ground for a very long time with a tremendous amount of stress and heat, will then add themselves one more neutron. And those carbon atoms and whatever composition, whatever thing that is made up with those carbon atoms will gain those neutrons and become that. And then we can read it. We've had the capability of technology in reading those number of carbon 13 atoms, and thereby, depending on how many you have, you can date something, right? So carbon 13 dating, for example, this is how we know fossils are X amount of millions of years ago. Does that make sense to you? Because of how many carbon 13 atoms it has in its composition, in fact. Okay, moving on. Molecules and compounds, right? This is just straight definition, right? But it helps us to understand when we talk about bonding, right? Molecules is when two or more atoms bond together. A compound is a type of molecule that is made up of more than one element. Okay? So, we would consider that. This is O2, by the way. That doesn't, not a weird, I should separate that, right? There you go. O2, right? Two oxygen atoms bonded together. And we create O2 or oxygen, two oxygen atoms, right? This would be commonly known as oxygen gas because it's in a gas state on Earth, at least. It makes sense? It's a molecule because it's two or more atoms, two there in that example. So what about this one then? Right, that's a compound. And why? It's still a molecule because there's two or more atoms there. There's actually three. There's two oxygen and one carbon. okay? But the reason why it's a compound is because it contains different atoms. One carbon atom, two oxygen atoms. Different, we call that a compound. And that's, of course, carbon dioxide. That makes sense, right? Simple. No big deal. Molecules are always written in a molecular formula. That's what we just saw right there. That's a molecular formula. In fact, those are all molecular formulas. And this one as well, right? Does anybody know what this one is? C6H12O6.
Glucose.
Yes, very good. Glucose. That is a glucose molecule or compound, in fact. That's what that is, right? All right, so there's rules for writing this. We're not going to practice these rules, nor are you going to be given problems to practice. That's more chemistry, but just a little background on that. So it's good to understand how this is written, but we're not going to really be testing, right, per se. Okay? So what's an organic versus an inorganic molecule? The key word or the key words are carbon and hydrogen. Okay? To be organic means that you contain carbon and hydrogen in your molecule compound, in fact, compound. If you have carbon and hydrogen, at least one of each one, then you're considered organic. Okay? If you have one and not the other, then you're considered inorganic. Or if you don't have either one, you're inorganic. Okay? So our example there is glucose, right? Or a carbohydrate molecule, right? Because it has carbon and hydrogen. It has at least one or the other. It actually has 6 carbon and 12 hydrogen, but it classifies as organic. And then inorganic, there's our example there, CO2, carbon dioxide. It has carbon, but it doesn't have hydrogen. And because that simple fact is presented there, then it's inorganic. Okay? What else is inorganic, right? Many of these compounds are inorganic, right? Look at this one. It's got hydrogen, but does it have carbon? It does not. It's considered inorganic. O2, 2 molecules of oxygen, that's not carbon or hydrogen. So it still falls under the inorganic molecules. Now in chemistry, if you were to take chemistry, there's 2 routes you can take, basically. If you want to study inorganic, that would be your general chemistry class, or inorganic chemistry. And if you want to study organic, you would study organic chemistry. There's rules between both of them. There's a lot more to discuss on one when you take those classes, or if you take those classes. Okay? What are functional groups? Functional groups are molecules, types of molecules with shared chemical properties. Okay? They're kind of, if this was a family, functional groups are very much like brothers and sisters, or very, very close cousins, first cousins. Does that make sense? Molecularly speaking, chemically speaking. So they're almost in the same, I guess, family. Very closely related. They might change by one molecule, one atom or 2 atoms, and that's as far as they've changed from one to the other. Okay? And so we name organic molecules by the functional group they contain. So here are the functional groups. You don't need to study these either. No functional groups. But just to show you that these are important biological functional groups. And if you're at a major in biology or biochemistry or one of those, nice, pretty majors like I did, I would have to learn all of these. And then there are modalities, or how they change and how they bond with other things and where you find them in the body. And so there's a lot more to talk about there. But this is what you see, basically. The R represents the part that changes basically between one cousin or brother from the other. See the R on there, right? That's the part that changes. So that means, for example, every single methyl group, single methyl cousin or brother has all of this here, except for the R. The R will change. R represents the thing that part of the chemistry that changes between one and the other. All right. Solvents versus solutes, right? Solvents are dissolving agents, and solutes are the things we dissolve into a solvent. Okay? Now, since we are talking about living organisms, most of our living organisms, their solvent is going to be water. Okay? For the most part, right? For the most part. Water is our solvent. And the things that we put into it, all the chemistry that goes into the water, including the surface of the molecule, the cell, or whatever, is the solute, the ions, things like that. Okay? And we call this type of solution that has water as the solvent an aqueous or aqueous solution. Just means that water is the solvent and everything else goes into there. Right? To break this mix. pH scale, right? pH is based on the number of hydrogen ions produced in a solution. Okay? And it's all based on, in fact, log base 10. That is math now. We're doing a little algebra-ish kind of thing. Log base 10 is whatever the number of things are times 10, and every single one is a tenfold, basically, up or down. So if you have one molecule, it's pH 1. pH 2 means you have 10 molecules. pH 3 means you have what? 100. pH 4 means you have times 10, 1,000, 10,000, 100,000, million, etc., etc. 10 million, 100 million, etc., etc. Those are molecules of hydrogen ions in a solution. That's what the pH scale tells us. Okay? I promise you, when we look at chemistry, you're going to it's going to be used later on, because we'll take a look at a lot of biochemistry and microbiology, because we're going to look at how structures are made in bacteria, for example, how they interact with each other and their metabolism. So we're going to use this for sure. Okay. Salts form when acids and bases react with each other. When we put an acid and a base together, and this acid and base solution, when combined together, forms anions and cations, or negative ions and positive ions. So when we put acids and bases together, what occurs is that we have separation of positive charged molecules and negative charged molecules, and we would call that a salt. In fact, a salt. So what are concentrations, right? The amount of solute dissolved in a specific volume of solvent. Okay? So in chemistry, if I added 1 milligram of whatever this is to 100 milliliters of solvent, we would just divide 1 over 100, and this would be 0.01. milligrams per milliliter. Okay? Very simple. This is how we do that. And then if we want to look at the percentage, it would be 0.001% concentration of this solution. Okay?
More acidic than pure water.
That's a good question. Does anybody know that? Right, rainwater's more acidic than pure water?
Because of runoff or something?
Well, yeah, because of runoff, right? Evaporation and taking water sources from evaporation up to the sky. And so there's different types of molecule, or I would say atoms in there, other than just pure water. You follow what I'm saying? Mm-hmm. Rainwater. And when it does that, you're going to increase the acidity level. Yeah, because of the other things other than water just in there. Yeah. It's not just H2O. It's H2O plus other things in there. You come from the evaporation runoff, and then that goes up back into the air. Yeah. Okay, here's logarithmic, log 10, base scale, right? There's a 10-fold difference in hydrogen ions for every whole number that we increase, right? From 1 to 2, from 2 to 3, 3 to 4, and et cetera, et cetera. We're multiplying whatever molecule is by 10 every single time, okay? So it's the easy way to do this, right? If it was 1 here, if it was 1 molecule of something, then 2 would say there would be 10, and this would be 100. And the next one would be 1,000. And you just multiply that number by 10 every single time, and that's the 10-fold increase, or logarithmic base 10, right? Just an easier way to multiply 10 to 10 to 10. If it's a constant 10, then it's a logarithmic 10. How we measure pH. Okay, and this is just breaks down, for example, the pH is 7. This is right at 7. This is 7 to 14. This is 1 through, this should be 8, right? Right? That's really what that means. The number, right, on the pH scale. Okay? So, where is this going to play a role, and why are we studying this right now? Well, because this right here is a great example of what you would see later on in the course. And we mean later on in like 2 weeks probably we'll see this. Okay? What we're seeing here is media plates, in fact, like the ones yesterday in lab, where you have media inside the plate, agar inside the plate, but instead we've added an indicator, which means there's a chemical in there, right, that makes this media, instead of that brown color you saw on the plates, a red color, right? And when we grow bacteria on it, which we're doing right now, right? So what they've done, what the person has done, is actually put bacteria on there. And what they want to do is see a color change. So this is how it comes normally, right? This is the normal color here, red. You see the red, right? Here is the indicator. The yellow is the indicator, which means as the bacteria is growing on the surface of the plate, okay, it is taking up nutrients from the surface, from the agar itself. Under metabolism, they're breaking stuff down so they can build more of themselves. A side product is made, and that side product interacts with the chemical in there, that's the red color, and changes the red color from red to yellow. Now that indicator is called phenol red. We don't need to know that right now, but phenol red is the indicator. And so if this was a plate and you're trying to figure out, okay, what type of bacteria lives on this plate or can grow on this plate, and that'll give you a clue as far as what it is, then it would change from red to yellow. You would know that that would be indicated of something. It would give you a clue to say, okay, it uses whatever's in here to grow, and because it does that, its side product changes it from red to yellow, or the pH indicator, because in fact it's making this more acidic, the plate more acidic. Because it's making it more acidic, the indicator changes it from red to yellow. Okay? Okay, so that was a little bit of a basic chemistry, but let's go into this, right? So this is more indicative of what we're discussing, right? Microbes, right? Usually, microbes grow best 6.5 to 8.5. That's its pH. Okay? Lo and behold, kind of right in the middle is what? 7, right? Neutral. So sometimes some organisms grow a little bit acidic or a little bit basic, but almost right in the middle of the pH scale. Does that make sense? Most microbes, not every single one of them on Earth, but most of them do. Majority of them do. Now, look at the pH of blood in the human body. This is this number down here, or this range right here, which you should know by now. If not, you need to burn that in your brain, because that's always something you will know if you're healthcare, is the pH of blood range, right? 7.35 to 7.45 is the range of our pH in our blood. Okay? Does that make sense? So even if we've got a organism that somehow got into our bloodstream, let's just use the in theory thing. Well, the pH of blood is what? Slightly basic, right? It's not acidic, it's basic on the basic end. 7.35, 7.45. Okay? Then most microbes can grow really well in blood, right? Just in pH. Just looking at the pH. Does that make sense? So we're trying to compare that in theory, if we were to have just blood, then organisms like microbes can grow in blood really well. The good thing is that humans and human bodies are built to withstand or block most, if not all, of those microbes from getting into the bloodstream. Now, there are situations where the organism will get into the bloodstream, which we'll talk about later on, right? There's ways to get into the body, but we've got a really state-of-the-art kind of advanced immune system, right? And since we got this immune system set up, that immune system pretty much blocks a lot of that stuff from ever getting into that part of the body. Okay? So let's talk about what acidosis versus alkalosis means. These are also medical terms, by the way, but it's also chemistry as well. When you have acidosis, that means the blood is actually lower than your normal range. That's 7.35, 7.45. So you can have acidic blood, and that's acidosis. Alkalosis is that number past 7.45, where the blood can actually be basic. We call this alkalosis. Now, there's different reasons why that would be the case, but our body also balances that out really well, and we do that through what we call buffers. A buffer is a compound, chemical, that actually changes the pH and balances everything out really well. Okay? All right. On to chemical bonds, right? All right. So chemical bonds are the glue or the attraction or the force that binds atoms in a molecule, right? So this is the molecular formula of, let's say, glucose, right, that I mentioned. That's the molecular formula. But in fact, there's bonds between all of those carbons to carbons and hydrogens and oxygens. See if I can draw this without messing up too bad. I didn't do too bad of a job. Okay? So it's a 6-ring type of molality. We call this molality, but it's a shape, right? And where the lines meet is where you're going to have a carbon atom in where the lines meet. The line represents the bond between carbon and carbon, and another carbon, another carbon, another carbon, another carbon. Call this a 6-ring type of shape, because this actually looks like this. Okay? Now, where are the oxygens? Well, I didn't draw that, but if I was to draw up like this and down like this, some of these would have oxygen. Some of these would have OH like that. Some of these would have hydrogen groups like that. And I would go on and on. It would start to become really messy right here, obviously, because it's really small, but this is how they are bonded together. So in reality, it looks like this. Okay? The line in between is the bond that we're talking about. Okay? Now, how do they bond together? In fact, they bond or glue together or are attracted together by the electrons outside of the nucleus of an atom. Those electrons are organized very separately from each other in what we call electron shells, which is not a shell, but we like to call it a shell because it very much surrounds the atom like or the center of the atom like a shell. Okay? Each shell, and I would call them little stages, either we'll see them in a second. Each shell has a maximum number of electrons it can fit in each one of them. And the outside shell, the outside electrons, is what we call the valence shell. Okay? The valence shell. So I want you to look at this really quick, right? There's 2 hydrogen atoms. The plus sign means that we're going to interact these with those and try to make a molecule. Right? This is a chemical reaction. Okay? The arrow in the middle means that we've forwarded the reaction. The reaction has occurred and this is the result, right? Basically. And what have we made with an oxygen atom? We've made water, right? In fact, which we know is the most abundant compound that we have on Earth. In our bodies, in fact, about 70% of our body is water, right? But look what's occurring here at water. Can we see that the electrons are basically in the same little circle, right, area? Which means they're interacting from one atom to another atom. The electrons are interacting, okay? Notice that hydrogen only has 1 proton and 1 electron. It doesn't have a neutron. 1 proton, which is in the nucleus, 1 electron. Oxygen has 8 and 1, 2, 3, 4, 5, 6. Okay? What it wants to do is actually grab 2 more to become 8. The rules of the chemicals interacting with each other. Okay, I'm going to come back to that in just a second. Atoms that are full have full shells, which means they have a complete shell, are stable. Okay? And that's a key word to understand because you have to be stable, okay? And every atom wants to be stable. That's its goal, basically, is to become stable. Once it becomes stable, it's going to live like that. It's going to hang out like that in nature. Okay? Now, in nature, if they want to fill their shell, they just don't grab electrons from anywhere. Electrons just don't flow around just kind of anywhere for you to pick and say, okay, I'm going to fill up my shell and I'm good. The only way you can fill up your shell is by interacting with another atom that has electrons that you want. Okay? That's the only way it's going to work. And on the same type of situation, but on the other side, that electron wants to share those or give those to the other atom for it to become stable. So it's got to be a relationship where they both are going to benefit. Does that make sense? Okay. In nature, there are some that are already full, like your inert gases or your noble gases. Now, we call these inert gases or noble gases because they are gas, they're in the gas phase all the time, number one. Number two, they're noble because they're stable. They don't interact with other things. They're kind of just by themselves. Okay? Helium is one of those. Helium is a noble gas that, for example, we can use to blow up balloons, right? That's helium. What about neon? Neon's also a noble gas, and that's something that if we add electricity to it, if we charge it, we shoot power through it, it starts to glow, right? Neon lights, right? Pink, purple, blue. Depending on what other gases are in there, how much electricity you shoot in there can change the color. Okay? The ones that are not full in nature are reactive. They're going to try to interact with other atoms. Those are the ones that we are going to study, right? How do they react? They can gain electrons, they can lose electrons, or they can share electrons between atoms. Okay? That's what we talk about when we talk about, because all three of these are types of chemical bonds. All three of those are chemical bonds. That's what we're going to discuss, right? Example that they've used here. This is salt, by the way. NaCl is sodium chloride, or your plain Jane table salt, right? And in table salt, if you take away the water, it becomes this crystalline kind of structure, this 3D kind of crystal cube, because that looks like a box, but it's actually a cube, right? With it, when it doesn't have water. When it has water, what happens? Water breaks the bonds in between the sodiums and the chlorides, which are positive, negative charged. Okay? The ions are free, and this would be salt water. Does that make sense? Salt water. You take away the water again, let's say you leave the salt water there and let it dry out, guess what you get back? Salt crystals. Yes, ma'am?
The water that says like it has electrolytes, what are they dissolving?
So let's just take an example like Gatorade, because Gatorade has a lot of electrolytes. In particular, it has potassium, chloride, sodium, and probably magnesium too. It's got all your electrolytes, just like that. So what they've done is they've taken all of these ions and they stuck it in water, and then they've added some coloring to it and then some flavor. That's what Gatorade basically is.
Oh, okay.
Yeah, that's what Gatorade is. Now, why do you need Gatorade? Gatorade was developed from I don't know if you know this, but from a scientist or from a doctor actually that worked at the University of Florida, right? The Gators, right? Because that's their, okay, yeah. And so he was on the football team, in fact. He was, treating his football players. Found that they were sweating a lot and cramping and not, they couldn't finish practice because South Florida is very hot, right? Sweaty, humid. So he said, well, let me try to find, because he knew that the electrolytes, because when you sweat your electrolytes out. So not only the water and sweat is water, but it's also all of these come out of your body, right? And when they do that, they affect your muscles and nerves can't function right and all that stuff. So what he decided to do is make this solution that he called Gatorade, aid from assisting gators, yeah. And Gatorade was what he formed, right? Made it lemon lime because that was the first one. And so that's what Gatorade is. Now Gatorade was bought by Coca-Cola, and Coca-Cola's, it's everywhere, and so you can eat it with your food, which is not the way it was ever designed. You never drink Gatorade by itself.
Yeah, unless you're like actually.
Right. If you're working out strenuously for a long period of time and sweating for a long period of time, Gatorade is helpful. Now, you can work out for 30 minutes and you don't need Gatorade at all. You can work out for an hour, and you probably won't need Gatorade. Now that's a difference between running, let's say, I don't know, 12 miles in July at 3 o'clock in the afternoon. You're going to be just dripping sweat. Like all of these are coming out really fast. You're going to need electrolyte replacement, and that's where Gatorade can be used. Or it's a strenuous activity like soccer or football or whatever. That's the only time you would probably need Gatorade per se. Does that make sense?
Yeah.
Yeah, that's usually what they do. But the problem is with that is that you add extra stuff that you don't need. And let's say you go to Whataburger and you want a Powerade with the big one like this. And you're adding what? Extra sodium also. That's more salt on top of the whatever burger you ate, salt, whatever salt, fries that had salt. Salt. It's a lot of salt.
So
Too much for both.
in that case, would drinking water with electrolytes be like natural water be bad for you since you really don't need it?
Like a Propel or something like that?
Yeah.
I wouldn't say it was too bad because the electrolytes are not that much in Propel, let's say. The sodium is the only one I would be concerned about. The other ones, we have a way of kind of balancing that out. The sodium too, we have a way in our body for kidneys to kind of excrete that. But sodium does some really interesting things with our blood pressure, and that's the issue there. Yeah. But Propel, I wouldn't say that was too bad of a kind of thing. I'm using Propel because that's like a vitamin water or whatever. It does have a little bit of salt. Depends on what kind you buy. Yeah, that wouldn't be bad. But on top of that, Gatorade has a lot of sugar, and you're using that sugar to replace your energy level. And that we know that sugar is already bad, and that's been an issue in healthcare. Okay. Covalent bonds is another type of bond, right? And covalent bonds, okay, is the attraction of atoms, and the key word is they share, okay? In our other example, in the ionic bonds, we gave away an electron, right? Okay, and the other one accepted an electron. We form two charges and they attract each other, and that's an ionic bond, right? We just talked about that. This one is different because covalent bonds are sharing. They're not giving away their electron. The other one's not receiving it. They're sharing electrons between themselves. Sometimes it's one pair. Sometimes it's more than one. Okay? But the key word is that they share. Covalent bonds are sharing electrons. Okay? Like this. These are two hydrogen atoms bonded together in a covalent bond. Now, one of these blue dots represents the electron for this one, let's say, and the other blue dot represents the electron for, let's say, this one, right? And we write it in the middle, we put the two dots in the middle to tell us that those two are shared between them, these two atoms. Okay? Look at this one, oxygen down here. Oxygen shares 4 between them, and that's why they write the 4 right down the middle. See that? It's because 2 electrons on the left, for the left oxygen, are getting shared by the 2 electrons on the right, and vice versa. 2 pairs, right? Or 4 total. Does that make sense? Now, in reality, these electrons actually continuously move around the atom. If my fist was the atom, the number 1 or the number 8 or whatever, okay, the nucleus, right? The electrons are moving around the nucleus. In fact, if you were to look at them and took I don't know if you could have the capability of technology to look at them and take let's say a video of it, you would see the electrons doing this. Does that make sense? And in fact, they're not doing just this because it looks like it's just kind of around like this. It's actually in front of you and back of you. It's a 3D kind of thing. It's going everywhere. And all of them are going everywhere. So there's 5, 8, however many, they're all going around and around and around. And in fact, those 4 there are going this way into the other one, and this way into the other one, this way to the other one. That's how they're actually moving in real time. Does that make sense? That's how they're That's what carbon does. Okay. A type of covalent bond, this is a sharing bond, we're still in covalent bonds, is a polar covalent bond. And what does polar really mean? They're sharing electrons, because that's what a covalent bond is, but this key word is unequal sharing. That means the interaction between one or more atoms results in the sharing of electrons, but one of the atoms is going to share more time with the electrons than the other atom or atoms will. Okay? They actually hold the electron with more and more of a given time than the other ones will. This occurs, for example, in water. Water is a polar covalent bond. And you can see in the example there in the figure that most of the electrons are pulled towards the bigger oxygen molecule. Okay? Now, these are two hydrogens, so they know that there's one of these is theirs, and the other one is theirs, right? One of them's to the left, one's to the right. All the other ones are part of oxygen. Those are all, they belong to oxygen. Okay? But you'll notice that all of these are pulled to this area. They've not gone over here, they've stayed over here. We call this a pole because one side and the other, like a North Pole and a South Pole, are two opposites. So one side of this molecule is going to be negatively charged, and it's this side. Has more of a negative charge on that end of the molecule. And the other ends are more positively charged. Right here. Okay? Because if we were to measure it over time, let's say we measure it for 5 minutes, most of the time is with the electrons are spent over on this side of the molecule versus that side. Because this is such a bigger particle, it pulls, or is like a bully, it pulls away the electrons and keeps it for themselves for most of the time. We call that a dipole. Okay? A dipole. Can everybody see that water has got that positive charge on the hydrogen ends right here and more of a negative charge on that end? Right? So much so that it starts to form other bonds with other molecules, and we call that a hydrogen bond. Right? Where this positive end is now attracted to a negative end of another molecule. And we call that a hydrogen bond. Okay? We'll come back to hydrogen bonds because we'll use hydrogen bonds here as well. Talk about them, right? So when we talk about van der Waals interaction, this basically means that when we have this polar covalent bonds forming in molecules, that attraction from one molecule to another forming these hydrogen bonds is what we call van der Waals interaction. Water itself does this with other water molecules. Okay? When we talk about substances that are hydrophilic or hydrophobic, we're talking about things that we put into water, and because water has this polar covalent bond type of characteristic, depending on the chemical nature of the molecule that we're putting into water, it can attract to water or it can repel from water. And we call that hydrophilic versus hydrophobic substances. Hydrophilic are things that are water-loving, that will dissolve in water, and hydrophobic are substances that do not interact with water or water-fearing. And it's because of their chemical nature. Okay? Just want you to understand the definition. We don't need to know substances that are hydrophilic or hydrophobic. We don't need to know that part. Here's our example, right? Probably an example that you've seen before, right? This is philic up here. And this is phobic down here. And so what are our examples? Sugar into water. You know that sugar disappears after water. You don't even have to use a spoon if you don't want to. You just let it sit there and then you don't see it anymore, right? For sure. Or hydrophobic, right? Like oil and water, which we all know that oil and water don't mix. We already know that to be true, right? See this in like salad dressing, right? Okay. Italian dressing, balsamic, things like that, where you have oil and water mixture, does this. Where the oil molecules are basically bonded together and then surrounded by water, right? They form these little bubbles, or these, we call these micelles, right? Where they have this kind of weird interaction where the oil molecules kind of just huddle up together and form these little bubbles, little clusters of oil. That's what you see there. All right, so that occurs as well. And so what does that mean to us in microbiology, right? Because we're talking about nice water and vinegar and salad dressing, but what does that mean to us, right? Well, when you look at something like this, this in fact forms the what? Plasma membrane of every living organism on Earth. What is the plasma membrane? The barrier of the cell, whether that be one single-celled organism like bacteria or multicellular organisms like humans. That layer there, or the plasma membrane, is made up of both hydrophobic and hydrophilic sides of one chemical. We would call this a phospholipid, yeah? Which is telling us that the head is a phosphate group, okay? It's a chemical attached to lipids, which are fatty acid tails. And that one side that points, the little circles that point out, are hydrophilic. They interact with water. But the inside is hydrophobic, fatty acid. The tails are. Which means water, which it says in water, can't easily go through the membrane, like the arrow represents going through. It doesn't really work that way. It does, but not really. Doesn't go easily through there. Nor do any other things that are hydrophobic. Okay? Which means that the organism can control what's going on inside here. Does that make sense? And can interact with what's going on the outside too. That's the whole point we're getting to, right? Because all of this is just chemistry, right? It's all chemical bonds. We'll see figures like this, like drawings like this, but in fact, that really means chemical bonds, right? We don't need to know the chemical bond, because this would be like a advanced microbiology course where we're studying all the chemistry behind it, but we just need to understand the basic chemistry to understand that this is actually all chemistry. Okay. So yeah, it takes a while for us to get to this point, but we're here at this point, the reactions, right? Okay. How chemicals interact with each other. Chemical reactions all involve making and breaking chemical bonds. Where chemicals have attractions to each other, let's say it's an ionic bond or a covalent bond or a hydrogen bond, and then we want to break those apart. Maybe we want to break those parts into little pieces. Maybe take those pieces and reformulate something or use it for something else. And that's in fact really how this works here, right? In a living organism. Okay? In a chemical reaction, reactants are the things that we go into a chemical reaction, and what we get out of it is the products. It's just two simple words to describe that. What is a catalyst? It's a substance that can be organic or inorganic, so it doesn't have to be strictly organic, it can be inorganic, that increases the rate of the chemical reaction. Or we commonly know that to be speeds up the reaction. That's abbreviated, RX, and in chemistry is means reaction. Speeds up the reaction. Actually, right there. That's what a catalyst does. Does that make sense to you? It's some kind of chemical that when added to the chemical reaction speeds up the reaction, makes it more likely to happen. Does that make sense to you right now? Okay. Because we're gonna add to that. What a catalyst is, we're gonna add to that. Okay, we'll come back to that. So these are simple terms, so I don't want you to get too confused on them. Okay? If you haven't noticed, if you've never taken me before, I try to break it down as much as simple as possible. Okay? And I use examples of it because that's kind of what I want you to do on your own. Does that make sense to you? When you're studying, you're going to have to make sure that you break it down to how you understand it. Okay? In science, many of these words are very complex and they got a nice term and it comes from Latin or Greek or whatever it comes from. But it really means very simple things. Okay? So in your notes are good to write, but they're not good to copy verbatim, right? You shouldn't make carbon copies of what you see in the PowerPoint. It should be, what does it mean to you? What is the example he's given me? Or whatever I'm writing. That's why I'm writing stuff on there. Because these would be my notes if I'm taking this class. Does that make sense to you? And what's going to be important is if you, tonight, what you should be doing, is looking over your notes and rewriting them. All right? Not copying them. That's a difference. Rewriting them, not copying them. That doesn't mean that you just take that, whatever you're writing, and just make the same exact copy again. It should mean what it means to you. It means this, reactions, this and that, the other. Because if you can understand it the way you can understand it, because it can be different from me and you, then you've got the material. Then you can hear a synthesis reaction. Oh, that means what? Making chemicals together. That's all that means. And then the word doesn't sound so daunting and it doesn't confuse you and you didn't have to memorize it over and over with a flashcard, synthesis reaction. If you understand it, then you can apply the information to whatever question there might be. Does that make sense to you? Okay. Because it's really important to understand that because you can write till your hands fall off because I already see somebody like, my hand, my wrist. It's true. I mean, you can write and write and write and then you're just like, my God, like running out of like memory and space and effort. Or you can write down what you think is important, right? Bless you. Okay, so back to synthesis reaction. What we're trying to do is build things up. Okay, we're building things up. And since we're talking about microbiology, we're building things in a single-celled organism. So every single living organism does this. They do synthesis reactions all the time. We do this. Bacteria does this, everything else in between. We build things with chemicals that we bring in, or chemicals we have in our body or in our cells already. Make sense so far? Okay? We combine two or more reactions or reactants together, and we're building things. Like, for example, we see this, right? You don't need to know that chemistry right there, but that's 3 moles of hydrogen 1 mole of nitrogen, and you actually made a new chemical. Or A plus B is AB, which means there's a bond created in between them. Right? New chemical. Sometimes it creates this, right? When we get AB, sometimes a side product is water. We create water as a side product. And that readily occurs in living organisms. Remember we talked about a catalyst right now? Something that we add to the reaction that makes something speed up, make it faster. Well, what we added is a type of catalyst here called an enzyme. And that's why it's sitting on top of the arrow. You see how this little crazy chemical name is sitting on top of here? That's the name for the enzyme or the catalyst that when we add it to this, made this much quicker. Does that make sense? That's all that means. We're just looking at how we can take A and B, and A and B can be as small as that, can be as complex as these, or even more complex. Now there's a step process in how these all combine. We don't need to know that unless you get into organic chemistry. Then there's a lot of that. You'll learn all the rules involved in how these two form and why they form and all that stuff. But we're just looking at the basic stuff. Now, a type of synthesis reaction, a type of reaction that we build things up, is called a dehydration synthesis reaction. And this is the one that when we add two chemicals together, it releases water. Water is given off as a side product. That's why we call it dehydration, right? Dehydrate means to remove water. Yeah? And in a reaction that's a dehydration synthesis reaction, what we've done is added this, right? A plus B, and made AB. And notice I didn't write it like this, right? I wrote it like that. Just to show you that this actually means there's a bond in between these two. Does that make sense? We made a bond there. We put them two together. We synthesized them. And why is it called a dehydration synthesis reaction? Because the dehydration means water was given off as a side product. Can everybody see the water? Like the arrow goes this way? Water's given off. Okay? And why do we talk about this? Because in living organisms, like bacteria and in humans and everything, many times our synthesis reactions are dehydration, where we give off water. Right? Why do you think you need to urinate occasionally? Right? Why do you feel the need to pee throughout the course of your day? One of the reasons why is because you give off water in all the chemical reactions that are occurring in your body. And water is a side product that builds up, and that water can be taken out of your body through our urinary system in our bladder, and we pee. Does that make sense to you? Okay. So what's a decomposition reaction? Decompose means to break down. Reverse the composition. Reverse making what? We break down. Or decompose. Now, we probably heard this word before when we talk about decomposition of a body or an animal, right? Rotting, if you will. That's a type of decomposition because chemicals are breaking down, which means tissue and cells are breaking down. But this also means this in the chemical sense too, right? Look at the AB, right? There's a bond there, obviously. And now we've got the products being A plus B, which means we've broke a bond or several bonds and we separated them. That's what you see here in this example. That's what you see here in this example. Now, in the living organism, usually we use water, simple water, to help break a bond like this, to then separate them. So it works really cool because it's opposite. If we create water when we synthesize, we can break it up when we use water, right? We make water or we can use water. We call that a hydrolysis reaction. or this word means actually two words. Hydrolysis or hydrolysis. Yeah, so okay, let's take this first one, right? Hydro means what? Water? What does lysis mean? Break or break down. Breaking water. That's what a hydrolysis reaction is. Can everybody see this one right here? This is AB. Right? Let's say it's AB. Oops. AB. Right? And we want to break that, and we want to create A plus B. We use water, and we break water apart because water is added to both A and B to separate them. Can everybody see this as H this is how water looks chemically, right? And what we've done is we've actually broken one of these bonds. And guess where this part goes to? Everybody see where it's at down here? Right here, yeah? Where's the H? The other H that got broken. On the other side. See how we broke water apart? Or we underwent hydrolysis or hydrolysis? See, when you break down the word and you look at the definition and kind of understand that, then guess what? This doesn't sound so crazy. What are we talking about? Hydrolysis, reaction, decomposition, breaking down. What does all this mean? Well, if you break it down to your own words, then this doesn't mean that much, right? Very easy. Now, an exchange reaction is more of a reality in the body. This is more how we actually do this in our body. We exchange, which means we swap types of molecules, compounds between themselves. So we take different ones and we swap. Can everybody see that A plus BC equals AC? That we've swapped, we've taken C and we put it here, and then we liberated B. See how that works? So what do you think is involved in this? Both decomposition and synthesis at the same time. So we'll break down and then we'll build up in one huge reaction. And that's what I mean by in reality, because in reality it works this way, where there's going to be exchange reactions. Okay? And then in chemistry you can talk about a single exchange or double exchanges where A and D get swapped and B and C get swapped. We call that double exchange, but that's really what that means. The activation energy is the energy required, okay? The energy required, the minimum energy required to get the reaction started. You need energy for reactions to occur in many situations in the living organism. Can everybody see that there's reactants here? And to break them apart separately, there are products there. It requires a certain amount of energy, and we call that the activation energy. You don't need to calculate activation energy, you just need to understand or know what activation energy means, right? Okay. Now, when talking about energy, we talk about two forms of reactions based on the energy that's involved. Okay? In an exergonic reaction, means that the reaction releases more energy than it spent to start with. So that means when a reaction occurs, energy is given off. We can measure the energy. It's given off. In an endergonic reaction, that means that the reaction uses more energy than it releases. So we have to put in energy, and a lot of it, more than when it releases. We have to invest more energy. So what does exo versus endo mean? Exo means what? Out or escape. Very good. That means energy out. Endo or ender means in, right? You have to invest more energy. Put it
You receive
in.
less energy, right?
You what?
You receive less energy.
Well, you have to put in more energy to the reaction. You have to put in more energy to the reaction. Okay. So let's get into this one, right? Talk about this really quick. Okay, and this is where I'm going to kind of breeze through, right? But I want to talk about the important macromolecules of a living organism, right? You can take carbohydrates and break it down into their simple building blocks, like the ones here. Take those simple building blocks and add certain things to it and make more synthesis reactions. And you can create, with carbohydrates, you can create proteins. Or you can take proteins and you can create carbohydrates if you wanted to. The living organism has the capability of doing that. That's what's really cool about these amazing molecules that every living organism uses in some form or fashion. Okay? So this goes on, and what I want you to concentrate on is look at the type of macromolecule, what it's made up of, what is its monomer or building block, okay? And what are their functions in a living organism? Okay, those four things. Does that make sense to everybody?
What was the last one?
What is their function?
Function.
They're like the function of each of those macromolecules. Okay? So we can move to the next chapter. And this basically talks about each one of those, but that's what I want you to pull out, those four things. Okay? Does everybody understand that? Okay. All right, so we'll stop here for this chapter.